spectrometry that is more sensitive, we can see that there are a number of co-eluting species with different molecular masses. So, what can we do in such a case? If the chromatography, even at several steps, doesn't give a full separation, we can change our approach and then go not for a separation in chromatography, but for a separation in mass spectrometry. As I said, electrospray mass spectrometry gives us molecular information. So for each of species with individual molecular mass, we have an individual trace. So then we go we can go for the more fine, isotopic fine structure. What is isotopic fine structure? Each combination of isotopes present in a molecule gives a mass. And this is the mass that is characteristic for a species. So if we can identify this isotopic fine structure, we can identify the species. We can give a molecular formula. So for this, we need very high resolution. So, for example, we would like to achieve a separation of molecules differing for a single electron, which has a mass of 0.5 millidaltons. So we go from chromatographic separation to the separation of molecular patterns, molecular peaks. And then when we think about this, and then when we compare the number of relative intensity of species detected, so for our elemental detection, the number of peaks that we detect depends just on the sensitivity of the instrument and then the resolving power of our HPLC system, our chromatography. But then when we go to molecular mass spectrometry, the number of peaks will also depend on sensitivity of the instrument and resolving power, but also on the ionization efficiency that will depend on the species and on the matrix, and then on data processing parameters that we are looking for selenized species according to different isotopes for different intensity ratios. So this is just the data process to achieve as low as possible error in mass measurement, because it will affect the number of putative selenium formulas that are proposed by the software. So of course the number is lower for lower mass. So just please have a look that for low molecular species we can accept mass errors even of 5 ppm. But then for high molecular species that becomes difficult, because it means that for mass 500 with low molecular mass accuracy, then we would need to check more than 100 different putative formulas proposed by the software. So these are these two parameters, resolving power and accurate mass measurements, that are essential for untargeted analysis of species. Of course, as you can imagine, such an experiment produces a lot of data. So I've already mentioned that we cannot process this data manually. We need softwares, we need powerful schemes for data treatment. So one of such possibilities is to create, a database a database of putative species, and then to match the isotopic pattern of the species with what we observe. So this is how it works in practice, that we see the pattern, so this pattern is not good, and then at some point of our chromatogram we observe, okay, this is what we expected, what is acceptable. So this probably is a selenium species, so now we can work more on the formula, on the structure. So this is one approach. Another approach, this is a paper that we recently published. This is another approach that goes just for 2 peaks in the isotopic pattern. And then again, using this scheme, we were able to find almost all species that were present, and then even without looking into seleno compound database. So it was a real untargeted analysis. Well, this is just the first step. The first step is the formula. So the formula gives us the number of different atoms present in our species, but of course we would like to know the structure. So the structure can be elucidated by fragmentation of the molecule. So it means that our molecular ion can be fragmented, and then if necessary, the fragment can be also fragmented. So, like this, we get the structures, first the formulas, and then the structures of the products. And like a puzzle, we can reconstruct our molecule. So this is an example how it works. This molecular ion was fragmented, and then it fragments again and again, so we call it MS2, MS3, MS4, and then from these fragments we were able to come up with the structure. So this was a newly identified species that we published like 2 years ago. And then using this scheme that I have just presented, we were able to resolve selenometabolisms of selenium-enriched microorganisms. So just 2 examples. One example of selenium-enriched yeast, which is the most popular selenium-containing supplement, organic selenium-containing supplement, and then also selenometabolome of lactic acid bacteria that shows nutritional and therapeutical effects. So why are we interested in this? Well, this information is necessary for understanding the selenium metabolism, the control of the enrichment processes in production of these food supplements, also to check safety and quality of the commercial products, and it may also help with the determination of the origin of these preparations. So just some data. This is selenometabolome of selenium-enriched probiotic. And then the main species that is present there is gamma-glutamyl selenomethionine. So this dipeptide. And then it is accompanied by 11 other species that we could fully identify, and 2 more for which the structure still needs to be elucidated. So these are the structures of the species that we found. And then there is a selenometabolome of yeast, and this is much more complex. This is until now we identify up to 150 species and belonging to different groups. And now what is of interest for us is also comparison of metabolism of selenium and sulfur by yeast, because of these 2 elements are quite similar. So then there is a question if selenium species are just the product of random substitution of sulfur by selenium in sulfur species, or there is a deeper reason why they are formed. There are some genetic mechanisms. And now I would like to move. I announced I will talk about microorganisms and animals. So the first part was, well, this is methodological introduction and some data about microorganisms, yeast, and this bacteria. And now let's move to our collaborative project with the University of Wisconsin, and about the synthesis of selenometabolites in animals. And the first project, initial project, was about turkey. So we studied selenometabolites in turkey liver, because what was observed that when we feed turkey with increasing doses of selenium, there is a quite large pool of low molecular selenium species that are formed. As you can see here, there is up to this more than 40%, around 40%, when we feed turkey with we supplement turkey with selenium. So we wanted to know the nature of these species, and then these are the data that we obtained. So what we saw that supplementation with inorganic selenium is accompanied by increase in both. These are high molecular species, looking at the beginning of the chromatogram. So these are high molecular species and low molecular species. But then these low molecular species are much more increased with the supplementation dose. So we wanted to know the identity of them, but of course there are no standards for this kind of work. So it was this untargeted selenium speciation, untargeted selenometabolomics, with dual detection to see, here we see elemental detection, so this is selenium, but we don't know the nature of the peaks. So this is just an example of one of the species, identification of this species with this structure. This species, this is selenosugar. Selenosugar linked to glutathione. So then this is the mass spectrum showing different fragments, fragmentation of this molecule, and then more detailed data about the fragments. There are masses, and this data allows to identify the structure of the molecule. So this was one of them, and here you have all of them. So this is what we were able to see. So all of them are selenosugar. So in these frames you can see the selenosugar part of the molecule, and then with different other molecules that are attached using selenium sulfur bond. So our conclusion was that we observed significant quantities of selenosugars with glutathione, cysteine, and methyl. And then these selenosugars were also found attached to proteins via sulfur-selenium bond. So these are the species. And then, as I said, we are interested in interdependence, intercomparison of sulfur and selenium species. And this led us to the second step of this project, this time with using rat as a model. And we this time we looked into selenium and sulfur metabolites, meaning low molecular species in, well, we started with liver of rat. So, I mentioned that selenium is a difficult element plagued with isobaric interferences. The same is the case of sulfur. So in this work we used a state-of-the-art ICP-MS instrument that allows us to form well, ions of sulfur and selenium with oxygen. It means that we avoid the argon-related interferences. So just have a look at the chromatograms, have a look at the data. In frame of this study, we tried also to compare sulfur, well, selenium, inorganic selenium, and selenomethionine. So there were two groups of elements. And then we studied sulfur, which is green, and two isotopes of selenium, blue and red. So when we see the distribution, so we see when we see the distribution and compare these chromatograms, we see that they are very similar with two exceptions. We have one peak that is absent in the liver extract of rats fed with inorganic selenium. This is this one. And then we have another one that is missing here but present here. So let's have a closer look at this. We go to molecular mass spectrometry and identify the species. At the same time, we compare the two chromatograms in order to account, well, to see if we can elucidate the identity of all the peaks. So in the case of rats, we see selenosugars, two selenosugars, one with glutathione, one with methyl group, and then we see some derivatives of selenoglutathione. So there are two groups of molecules that were found. And then we checked what happens when we increase the dose, what is the evolution, and then we checked at the same time sulfur species. So then, as you can see, there is no full, okay, so these are increasing doses, 0, 0.08, and then so increases in this column and then in this column. So selenium is red, so we see more and more selenium appearing. And then sulfur doesn't change, but then we have no real correlation between sulfur and selenium. So for the evolution of selenospecies, we see increase in selenosugars, and then at higher supplementation dose, we see derivatives of selenoglutathione. For sulfur species, it's pretty much the same. So even if we find sulfur, even if we find sulfur analogs of selenium species that are not dominant species in the case of sulfur. So these species are major in the case of selenium, but in the case of sulfur, they are minor species. So the hypothesis of biosynthesis of selenium species is a result just of a random replacement of sulfur by selenium doesn't seem valid. And now we are at this stage of more advanced studies involving genetically modified animals to see in more details what's going on, what is the mechanism of the expression of this species. And then what we observe that there is a specific mechanism of synthesis of selenospecies is supported by a recent work published in Nature just a few months ago, where there was a study of another selenium species, selenon, that was supposed to be just product of random replacement in ergothionein. And then what is the message is that there is a dedicated pathway of synthesis of this particular species. So we think that for these selenosugars that we observe, there is also a dedicated pathway. This is not just a random replacement. So this is an ongoing study that we are carrying out with researchers from the University of Wisconsin in the US. And with this, well, I would like to finish just with this summary, that developments in mass spectrometry make possible detection of selenium species on the basis of the selenium-specific signal. Then we can go for determination of the elemental composition formula based on electrospray mass spectrometry. And the same technique gives us a possibility to see the structure, so structural characterization. So then we can assess global speciation of low molecular selenium species, so-called selenometabolomics. And however, quantification is difficult because of the lack of standards. So we are still working on that. And well, for molecular mass spectrometry, electrospray mass spectrometry, there are differences in ionization efficiency. So the analytical response of species is different, so it makes it really difficult to evaluate quantitatively. And then in HPLC ICP-MS, just with elemental detection, we need baseline chromatographic resolution, and this is very difficult to achieve, that I showed already. And then there is the question of the biological role of the species, because for most of them we don't really know their biological role. So then and then so these are general conclusions. That despite the problems related to selenium properties and interferences, now the state-of-the-art mass spectrometers allow this sensitive determination. There is also a possibility to go for isotope ratio measurements that I haven't shown today. And this is a routine technique for targeted analysis when analytical standards are available. However, we need both techniques for exploratory, so untargeted untargeted analysis. And well, there is a hope that electrospray complemented by dedicated high throughput data mining may become a standalone method for selenium speciation analysis. However, ICP-MS still provides quantitative information and selectivity. So this was my talk. This is what I wanted to present it today. Now these are acknowledgements. People from our institute, Richard Lobinsky present here, some of the co-workers. Then the data come from one of our PhD students. This is the view from our city, yeah, the view of our institute. And then just to show where our institute is located, we are in the south of France, so France is here. We are close to the Spanish border. This is Atlantic coast. Mediterranean is quite far away. Here we have Pyrenees mountains. So this is this view, beautiful view. Then some famous people, two European kings born in our city, and then of course in France there is a nice wine produced in the area. So thank you for your attention.
Thank you, Professor. I think our participant must have been enriched with your interesting talks with very much high technologies, which actually been focused on mainly on the importance of selenium for health and the detection of selenospecies, determination and structural characterizations using HPLC, ESI, MS, etc. You have given a very lucid explanation about the elucidation of synthesis of selenometabolites also, both in microbes and in animals. Thank you, madam. And now I think the participant must be having some types of queries because they are very much interested in your talk. So I invite this session now for the discussion, that is interaction. So if there's any queries, kindly.
Yeah.
Hi, Zara. Nice talk. Yeah, I just want to ask one question. Selenium, which is selenium, which is present in the glutathione peroxidase 4, plays a very important role in protection against iron-induced death by protecting the lipid peroxidation. The question is, what is the maximum tolerable range of the selenium in our body?
Sorry, I cannot hear you clearly. What is the...
Okay. What is the maximum tolerable range of selenium in our body?
In the body, you mean in the tissue?
In the tissues, yeah. Which doesn't cause toxic effects.
Because then, what we say that is we reason in the daily intake. So, because in the body it depends, because as usual, when we go for any element, the maximum concentration will be in liver.
Okay.
Because then everything goes by liver. So then, the highest the concentration you will have in liver, and then these are. In terms of the dose, you have deficiency, optimal, and excess. So this is the recommended. So below 40 milligrams daily, it's deficiency. More than 400 is already too much.
Okay.
So then usually when you have selenium supplements, the pills that you take are at 200 daily. So then it's also the problem is that food supplements are usually taken by people who have a healthy lifestyle, good diet, and everything. So this is but usually, if you for supplements 200. But then in terms of tissues, the highest is in liver, and then because we also work a lot with supplemented food. So what we observe that, for example, in most edible tissues, like muscles of chicken or beef, even if we feed them with a lot of selenium, it won't stay in the muscles. It goes to liver, or if not, it's excreted with urine, and then it's gone. And then, because you say glutathione peroxidase is a marker of status, of selenium status of individuals. Now it's how to say. There is also selenoprotein P that is proposed as a new marker.
Okay.
So because then because it can assimilate more selenium, it transports selenium. So it means that if there is selenium in selenoprotein, there is selenoprotein P. It means there is something to be transported, and yeah. So this is,
Okay. Yeah, thank you.
okay.
Anyone? Okay. And next, I would like to thank my co-chair, Professor N. Mohandas Singh, and I also hand over to him for queries that he want to have from Madam.
Ma'am, thank you very much. It's a nice presentation, and I would say it's a very nice work. And what my query is that this is selenium and the sulfur is using in your very work, and what are the oxidations is more feasible to apply in this research work?
Well, excuse me, what's the question?
Oxidation numbers of selenium and
Yes.
sulfur. Is it the selenium is a 4 or 6 or 2 oxidation number, and then sulfur is a 2, or like which one is the more feasible in this work?
You mean the most in terms of bioavailability, in terms of health problem, in health?
No, it's a little bit towards the chemistry questions. What I mean is selenium valency or oxidation number. Suppose selenium positive charge 2 or 6 is more feasible, or 4 is more feasible, ma'am?
So then you mean here?
Yeah, yeah. For example, it forms the compound here.
Yeah, yeah.
With the ligands.
It's 2. Yeah.
Yeah. That time this selenium, no?
It's selenium
This
2.
oxidation
It's
number
2.
is how much?
2. 2.
2.
2. Yeah.
2.
2.
4 or 2?
2.
2. Okay. Somewhere I
But
have.
then with
Somewhere I have seen the 4 also.
yeah, because
Is
in
it
the
depends
feeding
on that?
studies, yeah. Okay, let's go. Because in feeding studies there is a big how to say, there is a big dispute if it's better to supplement selenium 4, inorganic selenium. So this, in a way, you know what you are giving to your patient, to your animal. Yeah. So then it means that this is a pure compound, inorganic selenium. But then now we go for green natural things.
Okay.
So if you want to mimic what is given, what is consumed naturally, so naturally you consume organic species. So we try to enrich food in selenium in natural way. So of course the easiest to enrich it will be yeast, for example, because it grows easily. So, and then in this work, what is this? We were just supplemented selenium 4. Yeah, this was selenium 4, because sodium. So because it's selenite. So then, well, the thing is that when you give selenium 4, you know what you give, and when you
Thank you, ma'am. After this, we shall continue with our oral presentation, OP12 to OP15. So may I kindly request OP12 to kindly come up on stage. OP12, Brojendra Singh.
The first technical session of plenary deliberation is over, and now we will go for paper presentations. And for this, I hand over the technical sessions to my to Professor N. Mohandas Singh, co-chair of this technical session. Thank you, Uda.
presenters, please, you kindly stick the rule behind. Presentation time is strict to the 8 minutes, and one question for the query. And now, this OP12, okay, this presenter is Brojendra Singh Sagol Sem, and his topic is the green synthesis of reusable, visible, active heterogeneous photocatalysts for water remediations. Presenter, please, your time. Please take your time now.
Hello. So, good afternoon, respected chairman, co-chairman, resource person, and other my dear colleagues. I myself, Brojendra Singh Sagol Sem, I am doing research under the supervision of Professor N. Mohandas Singh sir in the Department of Chemistry, Mizoram University. Chemicals is nothing, it is the substance that can alter the rate of reaction without being consumed in the reaction. And if the catalyst is activated by using sunlight, then we used to call photocatalyst. So here I'm going to focus on photocatalyst. And the photocatalyst, here ternary photocatalyst, we are going to prepare by using ferrous ferric oxide, tin dioxide, and graphitic carbonitride in the presence of plant extract. So here lemon I'm using, from this plant extract I'm using preparing heterogeneous photocatalyst. And to prepare this ternary photocatalyst, I'm using this one hydrothermal methods. Hydrothermal method is a chemical reaction in a closed vessel that is called autoclave, in which reaction takes place at high temperature and pressure. So these are the steps that I follow to prepare heterogeneous photocatalyst. So this is the design of compound. Here ferrous ferric oxide is there to have the magnetic property, and the others, tin dioxide and graphitic carbonitride, is to excite electron, and so that we can separate electron and the holes, and that can use for the degradation of dyes. So here's the reaction mechanism. So I already explained in the previous slide also. Here pollutant plus holes will form oxidation product, and the pollutants plus electron will form reduction potential, and that always generated during the degradation of dyes, that will help for the degradation of dyes. So for the characterization, I am going to use UV visible spectroscopy to find out the band gap energy of synthesis compound, and another is to find out the change in the concentration of dyes with irradiation time. And the second is XRD to find out the morphological structure, FTR to find out the functional groups, and here is the tau equation. Yeah, by using this equation we have calculated band gap energy, and here is the band gap energy for ferrous ferric oxide, that is 1.7 electron volt. So like this we have calculated for ternary photocatalyst also, and we calculated band gap energy here by using tau equation. So now FTR to find out the functional groups. Here band at 524 centimeter inverse represent the ferrous, means iron and oxygen banding, and the 524 is oxygen, iron oxygen banding, and from 80 to 1396 is for graphitic carbonitride, that is fingerprint region for graphitic carbonitride, and 3302 that is OH stretching. Now XRD. So this XRD spectrum I compare with the JCBDS, and all are I found matching also. So it agrees that we can prepare this compound successfully from this graph. Now that, for XRD data we can calculate the grain average size, and by using Serrer equation. Here is the Serrer equation. So by using this Serrer equation we have been calculated crystalline size. Here D stands for crystalline size, K is the grain shape factor, lambda is the wavelength of X-ray, beta is the line broadening at half maximum intensity, and theta is the Bragg's equation. By using this one we have calculated the crystalline size of each and every compounds, and this is matching with this reference number. So here is the same image for this ternary photocatalyst, and it has mesh shape. From this we can see, and the next is EDX from SAM. So from here iron is from ferrous ferric oxide, and the tin is from tin dioxide, carbon and nitrogen is from graphitic carbon nitride and oxygen from its oxide. So it confirms that we prepare our sample nicely. So RSB standard curve is, I make this one by using different concentration of dyes, that is 2 ppm, 4 ppm, so on. And what is the purpose of doing this is we are going to do degradation so we can find out its concentration if we know the absorbance. So if we know the absorbance value, then corresponding concentration we can find out from this standard curve. So for future use, I'm prepare this one. So dyes concentration effect here, I use different concentration of dyes, that is 2 ppm, 4 ppm, so on, and by using 1 milligram of ternary photocatalyst. So from here we found that 8 ppm has more degradation efficiency, that is 93.60 something. So we can conclude that it has more effective in 8 ppm of dyes concentration. Here RSB means rhodamine. I am using as model polyethylene for this purpose. So from that equation data, I calculated Langmuir and the Findley's isotherm. So we found that R square value of Langmuir isotherm is 0.98 something, and Findley's equation has 0.78 something. So here R square value is very close to 1 in case of Langmuir equation. So we can confirm that it follows Langmuir equation. Now, from that same data, we calculated pseudo first or pseudo second order. Here also R square value in pseudo first order is 0.92 and pseudo second order is 0.69. So here also R square catalyst, it also affects the dissociation of dyes also. So there is side effect of OH in this case. So it has different spectrum. Now this is for this one acidic medium. So in the acidic medium there is a large hydronium ion, so that will generate peroxide, and from there more hydroxy will generate. So that will deform the degradation. So this is the kinetics of energy at different pH, and after considering all the effects of OH, it is assumed that pH range from 6 to 8 has more efficient than as compared to others. This is the separation. By using external magnet, we recollect the catalyst what we use, and that is used for future. So here is the VSM. VSM is vibrating separator to measure the magnetic property of the compound. So here iron has more magnetic property as compared to others, and others due to impurities it has less magnetic property. So now we can recycle it at least 5 times without much difference in its degradation. So we can use 5 times successfully. Here is the conclusion. We can prepare this ternary compound successfully in the presence of plant extracts, and it has more efficiency than binary. And we are doing this in the sunlight, so it is eco-friendly. And the next is we are using external magnet to collect it again. So it has more advantage than other normal filtration and the centrifuge, because in the normal filtration and the centrifuge filtration, there is deformation of your compound. So it has more advantage. And the last is it can be used as a photocatalyst with great performance in the degradation of dyes. Now this is a reference. Thank you for your kind question.
Thank you, Mr. Brosen, for the nice presentation, I could say. And now open for the audience. Only one question. It will be more better. It looks like the conference or seminars. Okay. I do hope no questions from the audience. And thank you, Mr. Brosen, once again. And then next presenter was I'll invite the next presenter, Singhabam Dhananjoy Singh. Topic is the scrub typhus in Ukhrul and bacterial identification. The rule is the same, huh? 8 and then 2 minutes for the interactions. Yeah, stage is yours now. Okay, thank you.
Okay, so my background is scrub typhus in Ukhrul and vector identification. So background, scrub typhus is prevalent in Manipur, as we know, and this is by a febrile disease without any symptoms. Hello. Okay. So scrub typhus is a febrile disease without any specific disease-specific symptoms, signs and symptoms. Usually, it has symptoms of headache, body aches, this one fever, and rust. Sometimes it has a scar. This is caused by Orencia susugamusi. It is a rickettsial bacteria, and this bacteria is transferred to human through the bite of this cigar of trombocolid mites. And if it is not treated well or late, it is a fatal disease. Sorry. So this is a scar from our patient in district hospital, Ukhrul. Historically, it was mentioned, this scrub typhus was mentioned in 313 in China, and later on in different parts of the China reported. In 1810, it was reported in Japan. Then the name Susugamushi comes up from Japan, and in 1932, India report the first record of scrub typhus. This is concentrated in Susugamushi triangle, mainly the northern part of Australia, Japan, India, Afghanistan, all this. But due to the development in transportation and tourist, the location of this scrub typhus is expanded from this Susugamushi triangle. In case of Manipur, please avoid these areas. Okay, then if it is infected, then you do take this one fever. Okay, then treatment is doxycycline, only doxycycline. In case of pregnant woman, you should be azithromycin. Okay, so once again, I like to focus one. Okay. Okay, so in case Manipur, most of the hilly districts of Manipur is infested with this scrub typhus bacteria. Okay, so whenever you go there, you test it. Okay, so in the Ukhrul, the test is 853, out of which 75 is positive and 59 is emitted, and still till now 7 had death. But in my study period, it was only four deaths. In Kampopi, three deaths. In Infant West, it was no death, but Infant East, it was 1 death last year. Okay. So Ukhrul has a population of 2 lakhs, nearly 2 lakhs, and having 4 blocks administratively. And Ukhrul block has 1 million, 60% or 1 lakh. 1 lakh. And 60% of the population is concentrated in Ukhrul district, Ukhrul block. So this scrub typhus was first discovered in 2016 by our DSO Thodaichon, and in 2020 it was very popular in Ukhrul district due to 2 deaths in Khamasom Phungdar, and later on in 2021 it was reported in our reporting system. So objective of the study is to perform descriptive epidemiology.
Thank you, Oza. Keeping more time. It's less than 7 minutes. And I could say some words is that this has already been published in the local newspaper or?
No yet, sir.
Okay, if it is there and it gives to the layman peoples,
yes,
more interactive.
Yes.
Okay, now open for the audience. At least one question. If anyone has interest on this topic.
Okay, so this scrub typhus is more prevalent in Ukhrul district because most of the farm works traveling and everything are mostly done by the females.
Okay, one more.
Pardon? Okay, sorry. Let me go to this slide again. Okay, so in Manipur, this scrub typhus is prevalent in most of the hilly areas of Manipur, hilly districts of Manipur. You can see Kampopi last year 3 deaths, Chuchampur 1 dead, Chandel also having that one, but not reported in our reporting system. But in Ukhrul, I managed that one, so 7 deaths till now. But in my duration of study, it was 4. So scrub typhus, whenever you people go to Ukhrul or any part of the hilly districts of Manipur, you take care, okay? Because you wear this one, preventive measures. You should be. Oh, okay. Sorry. Okay. So some chemicals are also available in market. You pass it around your body whenever you have a trek in mainly the hilly districts, not all. Okay, but this scrub typhus is more profound in Ukhrul district.
Yeah, thank you very much, Mrs. Speaker. And I do hope this will be published in the local newspaper, or possible in the very nice review, I could say. And thank you for nice presentation. And
Thank you.
yeah, and next. Diversity in India. This company also reviewed the presentations. Yeah, please. Yes, slide please.
Good afternoon, everyone. Good afternoon, everyone. Thank you for the organizer and the sponsor.
Okay, time starts now.
Good afternoon, everyone. Thank you to the organizer and the sponsor for organizing, for providing an opportunity of my presenting my paper. At this conference. Today's my topic is biodiversity in India. We have already known that biodiversity terms and what are its compositions. Biodiversity means a variety of the life on the earth, and according to United Nations Earthsmith 1992, biodiversity is defined as the variability among the living organisms from all sources, including terrestrial, marine, and other aquatic ecosystem and ecological complex. The other part include diversity within species, between the species and the species and of the ecosystem. Then India is recognized as one of the mega diverse country, rich in biodiversity and associated traditional knowledge. Biodiversity is a part of nature which includes the difference in genes among the individual of the species and variety richness of all the plants and animal species at different scales in space, locally in region of country and the world, various type of ecosystem, both terrestrial and aquatic, within a defined area. As biodiversity is classified as genetic diversity, species diversity, and ecological diversity. First of all, the genetic diversity. In the genetic diversity, each member of any animal or plant species differ widely from other individual in its genetic makeup. Next, species diversity. The number of species of plant and animals that are present in the region constitute its species diversity. This diversity is both in natural ecosystem and in the agricultural ecosystems. ecosystem diversity. There are less number of variety different ecosystems on Earth which have their own complement of distinctive interlinked species based on the difference in the habitat. Ecosystem diversity can be described as the specific geographical region or the political entity, such as the country, state of Taluka. Distinctive ecosystem include landscapes such as forest, grassland, desert, mountain, etc., as well as the aquatic ecosystem. Yeah, biological diversity classification of India. Our country can be conveniently divided into 10 major regions based on the geography, climate, and other patterns of vegetation, since the communities of mammal, bird, reptiles, amphibians. Then these are the Indian biogeographical zone, the cold mountain snow-covered trans-Himalayan region of Ladakh, the Himalayan range and valley of Kashmir, Himachal Pradesh, Uttarakhand, Assam, and the northeastern state, the Terai lowland where the Himalayan river flow into the plains, the Gangetic and the Brahmaputra plains, the desert of the Rajasthan, semi-arid grassland region of Deccan Plateau, Gujarat, Maharashtra and Andhra Pradesh, Karnataka, Tamil Nadu, northeastern state of India, western Ghat of Maharashtra, Karnataka and Kerala, the Andaman and Nicobar Island. Last one is the long western and eastern coastal belt with sandy beach forest and a mangrove. Values of biodiversity. Environmental service from species and ecosystem are essential at global region and the local level. Production of oxygen and reducing carbon dioxide, maintaining the water cycle, protecting soils are important services. Then important services. Yeah. Such as fishing of recycling, nutrients, soil formation, circulation of cleansing of air and water, global life support, plant absorb carbon dioxide and give out the oxygen, maintaining the water balance with ecosystem, watershed protection, maintaining stream and river flow throughout the year, erosion control and local flood reduction. Modes of conservation, ex situ conservation and in situ conservation. In conserving biodiversity outside the area where it is known as the ex situ conservation, here animals are reared or plants are cultivated like zoological parks. In situ conservation, conserving the animal or plant in their nature is in situ conservation. The established natural habitats are national parks, sanctuaries, biosphere reserve and reserve forests, protect forest, natural reserve. Next conclusion. Oh no. The great variety of life on Earth has provided for man's need over thousands of years. This diversity of living creatures from a support system which has been reused by each civilization for its growth and development. Those that use this bounty of nature carefully and sustainably survive. Those that overuse or misuse it disintegrated. And science has attempted to classify and categorize the variability in nature for over a century. This has led to understanding of its organization into communities of plant and animal. This information has helped in utilize the Earth's biological welfare benefit of humanity and has been integral to the process of development. This includes better health care, better crop, and use of this life form as a raw material for industrial growth, which has led to the highest standard for living for the development world. However, this has also produced the modern consumeristic society, which had a negative effect on the diversity of biological resources upon which it is based. The diversity of life on Earth is so great that if we use it sustainability, we can go on developing new products from biodiversity for many generations. This can only happen if we manage biodiversity as a precious resource and prevent extinction of the species. These are the references which I have collected from this book. Thank you.
Thank you very much. Any query from the audience?
So, what will your finding?
Sir, pardon me.
If you have any finding,
No, I just collect only, yes. Review only, yes.
My suggestion is next time, please try to get at least some outputs from your own work. I'm not discouraging you, and so that the moment you have done the things, even biodiversity, you can go around and collect like previous speaker have done. I was very much intense in this work with the Professor Rasmohan during 2009. 8-9 now we have done a couple of works. Very nice. Thank you, presenter. It's a very nice one. Yeah. And last, not this is the last presentation for this OP. Devika Chorokaibam. Topic is total phenolic contents and the reducing power assay of nymphaeic species.
Hello, good afternoon to everyone, respected chairs, co-chairs, teachers, and all my dear colleagues. Today I'm going to, I am Devika Chorokaibam, a research scholar in the Department of Botany. So today I'm going to present a topic on total phenolic content and reducing power assay of Nymphia species. Actually, this is a water lily. So introduction is, so while edible plants.
Please leave the introduction instead. You go to the result discussion.
So in biological classification, it belongs to the family of Nymphaceae, and genus is Nymphia. Short morphology is flower is numerous petals with spiral arrangement. The leaves are rounded and waxy coated. Stem is fleshy stem and the horizontal creeping rhizome present. And the sample collection, I collected the sample from Loktak Lake during the period from June 2021 to December 2021. This purple water lily, also known as, in common language, it's known as Tharoa Sangba. Here are the parts of the plant collected. This is our flower, stem, and rhizome. And the red water lily, in common name, it is known as Tharo Angangba. It is, I collected it from Ishokpat. And the parts of the plant collected are flower, the stem, and rhizome. So my main objective is to determine the total phenolic content and reducing power assay of the flower, stem, and rhizome of both purple water lily and red water lily. And the determination of total phenolic content was determined. First, I prepared the methanolic extract, and I again collected the extracted by using Whatman filter paper 1. And the total phenolic content of both the extract of flower, stem, and rhizome of water lily, purple as well as red water lily, was done with Folin CO-Cal2 reagent method by Singleton and Ito. And here in this total phenolic content of the three parts of flower, stem, and rhizome, it was found that the stem has the highest total phenolic content and lowest in the case of rhizome. Similarly, in red water lily, that is Nymphia rubra, the highest was found in flower and the lowest in stem. And in conclusion, it can be concluded that the total phenolic content was found the highest in the stem of Nymphia pubescens and the highest in the flower of Nymphia rubra. So we can conclude that there is a significant difference in total phenolic content among the flower, stem, and the rhizome. And this is for determination of reducing power assay. I determined it by following the method of Owizu, 1986. It shows that increasing in reducing power was indicated by increasing the absorbance of the reaction matter. Actually, this reducing power assay is a method where substance having reduction potential with potassium ferricyanide was transformed to form potassium ferrocyanide. It further reacts with ferric chloride to form ferric ferrous complex that has a maximum absorbance of 700 nanometer. So the result of my work is that by comparing the absorbance which I get from different concentration of both the standard as well as the sample extract, I found that the standard increases with increasing in the amount of the sample as well as the standard, and that shows a good linear relation in both the standard and the sample extract. And the same result was found when I compared the data between the standard ascorbic acid of flower of Nymphia rubra along with the sample extracts of the Nymphia rubra. There I only compare it with the flower because it has the highest concentration. And in conclusion, we can find that the antioxidant activity highest in the rhizome and lowest in the stem of Nymphia pubescens. Therefore, it can be concluded that reducing power assay of the methanolic extract of Nymphia pubescens and rubra, and was as the standard increases, with increase in both the sample and the standard. This shows that the reducing power has a good linear relation between the standard and the sample extract. And these are the references. So I would like to thank my gratitude to my supervisor, Professor Kanan Balasarangthem, and also thank the organizing committee of ICM BBH. And lastly, I want to show my appreciation and cooperation for my lab maths teachers and non-faculties of our department. And thank you, everyone.
Thank you, Ms. Speaker. Now open for the audience. Any query from our speaker? No. And then, if there is no question, I will ask one question. Now, you said that this is a phenol content.
Yeah.
How you have done characterization about that? Characterization to indicate that this particular, your flower contains the phenolic group or phenol.
Actually, I have done the methanolic extract first, and then I do a series of extraction using Waldman filter paper and methanolic extract. First, I made that.
And then how you can confirm that one? Can you tell me little that analytical part you have performed during your work, your lab work?
From the methanolic extract, I conducted an experiment using the Folin Culture method by Singleton and et al. And I prepared a series of dilution of the standard as well as of the sample extract. And
really, this thing you have taken from the literature, might be.
Okay.
Okay,
Yes.
and the conclude is that the phenol content the particular flower, plant. I do hope. And if you are doing really, you have to go for some other instrumentation, isn't it?
Yeah.
Like your mass and NMR.
Okay.
Without that, one cannot say this is a phenolic compound. Okay.
I estimated the absorbance by using the UV visible spectrometer.
UV is nothing. Okay. Yeah, that is just telling about the peaks. Doesn't mean, okay.
Okay.
And please you go a little in depth. Maybe that paper will be more impact.
Okay.
Okay. Thank you very much once again. Yeah. And I hand over.
Thank you. Thank you, Oza. And with this, I think we have ended the technical sessions. We had one plenary lectures delivered by Professor Jonas Spooner, University of Pau, other reasons, Pau, France. And we have an oral presentations of OP 12 to 15. And all the deliberations were done very well, and I think there was some interactions, and I think the participant must have gained something from this presentation also. With this, I would like to thank our resource person, Professor Jonas Spooner, and also Professor N. Mohandas Singh for co-chairing this session. And with this, I think we end this session. Thank you all.
Thank you, ma'am. May I now request Professor T.H. Binoy Singh to kindly felicitate Professor Kananbala. And I also request Dr. Birla Singh to kindly felicitate Professor N. Mohandas Singh. A big round of applause, everyone. Thank you, sir. Now that is the end of our oral presentation. So now we shall proceed with the young scientist oral presentation, which will be chaired by Professor Joanna Spunner and Professor N. Mohandas Singh. So the young scientist oral presentation will be from OYS 11 to OYS 33. May I repeat? OYS 11 to OYS 33. So there will be a parallel session on the venue in the conference hall. So that would be from OYS 34 to OYS 42. OYS 34 to 42 will be in the department conference hall. So those who are going to give the presentation in the parallel session, you may head to the conference hall. Go great. I hand over the session to the chair. Thank you.
So then it will be 11. Okay, so welcome to the session of young scientists. And then the first presentation will be number 11 and will be given by Samandram. Yeah.
What's her name?
11.
No.
11. Oh.
Sushil Kumar Singh.
I just realized one thing, that to be volunteer is much better than to stand up here. A very good afternoon to one and all present. Myself, I am Samandram Sushil Kumar Singh. I am a research scholar at the Department of Zoology, Manipur University. And today I'll be presenting on the topic phylogenetic relationship of Mylonculus lacustris with other species of mononchids from Mizoram using molecular marker. So when we say nematode, the ultimate example that we come to our mind is that of Ascaris lumbricoides, then we have loa, then Wuchereria bancrofti. These are animal nematodes that causes a large number of clinical manifestation to the human beings, causing a large number of diseases. So little do we know that nematodes do exist in plant and water also. So basically we see as a broad level, nematodes are economically important to the human being, both as pest and parasite. So today I would like to take everyone present here to the world of the nematodes present in the soil. So when we say mononchid nematodes, they are a group of plant nematodes or soil nematodes present in the soil that are microscopic and predaceous. From the groups of the nematodes inhabiting in the soil, they can be identified by the characters of its robust body, a well-developed feeding apparatus which consists of buccal cavity and muscular pharynx, caudal glands, and spinneret. Now, looking at their feeding behavior, mononchid nematodes feed on bacteria, nematodes, rotifers, and planktons present in the soil. So, because of their feeding behavior, on the nematode, these mononchid nematodes are used as a biocontrol agent of the phytophagous nematodes. Now, the term that I have used is phytophagous nematode are those kind of nematodes which are present on the soil and parasitize the plant. So, the aim of my study is to see the relationship between all the genera and all the family of the other mononchida, which was provided at the latest in the year 2010 by Ahmed and Zerasbury. So to do that, what I have done is I have collected soil sample from different parts of Mizoram, which was processed. Then nematodes were isolated. Then I have used light microscopy imaging and molecular studies. So for the soil collection, I have collected the soil sample from a depth of about 5 to 15 centimeter, depending on the plants. And I processed the soil sample using the scope sieving and decanting method, followed by the Beerman's funnel technique. And for light microscopy, I have fixed the nematodes at formalin alcohol solution and followed by the slow dehydration. For the molecular characterization, I have isolated the DNA using 20 micrometer of worm lysis buffer, 1 microliter of proteinase K, and centrifuge speed of G-force of 14,000. And isolated DNA sequences were amplified using the primer called as the SSU18A and SSU26R. So for the phylogenetic analysis, I have used maximum likelihood tree based on the 18S ribosomal DNA sequences. Now, to make the tree, I have uploaded 10 sequences obtained from the mononchid nematodes, which was obtained from Mizoram, and 45 sequences of the sequences were downloaded from GenBank and CVI. After that, the sequences were aligned, and the tree were constructed using the Tamuranai model. So, after thorough analysis or investigation, what I found is that I found 10 mononchid nematodes from Mizoram, of which all of us report from Mizoram, except this Brionculus cirroi, and all the sequences, including that falls under the Ionthunkidi, are all new to the science. So I'll not be explaining all the 10 species because I don't have the time. So I've just selected one species, that is the Mylonculus lacustris. We have the images. Now this species can be easily identified by its small body. It has a funnel-shaped buccal cavity. It has a large, massive claw-like dorsal tooth, and it has this raps-like 7 rows of raps-like teeth. So this is the dorsal tooth. And if you see the female characteristic, females are characterized by presence of this vulva. Sorry. So sorry. Female are characterized by presence of this vulva, and what I found is that the female here, they have this amphidelphic reproductive system, and I also found an egg which was in spherical shape. If you see the tail are cylindrical in shape, coiled and hook shape, and the caudal gland are in tandem shape, and the spinneret is in terminal. Now this is some morphometrics I would like to skip. Based on the molecular characterization, I was able to generate an 852 base pair long 18S ribosomal DNA sequences from the Mylonculus lacustris, and it was uploaded in the database of the NCI, and I was awarded with this accession number. After the NCI blast performed, what I found is that the sequence that I have uploaded has 100% identity with the Mylonculus species, which was reported from Iran. Also, it has a 100% identity with 98% query cover with Mylonculus species, which was reported from Taiwan and some other sequences. So this is the phylogenetic tree that I have drawn. Here we can see that A represents the phylogenetic relationship between all the family of the other mononchida, and here I have just condensed the tree to its and every clade that I have found. Based on the tree that we found is that mononkit nematodes, they can be grouped into 6 clade, including the outgroup, which I have used as a outgroup. So each and every clade consists of a family or 2 families. So I'd like to skip just to save time. So this is just the morphological characteristic that I have used in the genera for the tree. Again, this is the continuation. So a conclusion is that Mizoram, being part of the Indo-Burma hotspot, is definitely rich in the mononkit diversity. Again, what I found is that from all the mononkit diversity that I have recovered from Mizoram, Mylonculus genus is found to be the most commonly observed. And the start of the aim of my study was to develop a phylogenetic relationship between the different families of the other mononkita, which I was able to perform. And I also found out that the classification provided by Ahmed and Zeraspori back in the year 2010 happens to agree with the new tree that we have provided. However, there are some unresolved clustering in the tree, as that of the positioning of the Prionculus Alexandri in clade A, polyphyletic origin of the Anantoncus tridentus in clade 2 and 3, and the positioning of the Jansenioncus and Malleolus in clade 4. Beside these 3 unresolved clustering, all other genera, all other sequences of the mononkit nematodes fall under the classification of this Ahmed and Zeraspori classification. So yes, we understand that 18S ribosomal DNA sequence is a conserved sequence, but in order to solve all this crisis, all this unresolved clustering in the diversity in the classification, what we need is we need large number of 28S ribosomal DNA sequences and a large number of mitochondrial DNA sequences, which we don't have the luxury of that at the moment. Now, there are many sequences for these 28S ribosomal and mitochondrial DNA sequences for other nematodes, but unfortunately for mononkit nematodes, there are very few sequences for these markers. So what we have to do, what we have to look for, what is that we have to create more sequences based on this 28S ribosomal DNA and mitochondrial DNA. So what we have discovered so far is just a tip of an iceberg. We need to explore the area, we need to explore the biodiversity so that we get the maximum number of mononkit diversity. We have to look forward to our goal, and we have to take a leap of faith so that we trust in our goal, and we definitely will achieve our goal. So before I say thank you, I'd like to acknowledge all the individuals on the slide, and thank you.
Yeah, 2 questions from audience. No, ma'am uses this.
No, I don't.
Okay, ma'am doesn't have the question, and my query is, you have been applied for the patent.
No, sir.
Your work is, you have discovered any kind of this,
Yes, I have discovered, I have already published a new species. From this also, I have a...
When you have done these things? My question is,
pardon, sir?
When do you complete to find out all these species?
Sir,
2 months before, 1 month before?
So this is a study for a collection of based on 2 or 3 years. So I cannot just simply publish a paper. When it comes to taxonomy, we have to look for many things, like we have to look for the identification.
No, if you are slow, then you will be the loser, right? Over the world, many researchers are keep touching with this work. My suggestion is, isn't it?
Yes, sir.
And if you are delaying, then other people have discovered, then you are useless.
Yes, sir. Actually, sir,
and yet to publish, he's saying that. Yeah, means that he will be the loser, no?
So actually, sir, this one...
It will take time, then other people have found somewhere others, and he will be the loser.
No, sir,
no meaning for the research, right?
I already have a new species published, sir, and this one, this here, actually, just recently 2 days back, I have just completed the review of the paper. It's about to publish.
Probably you try to apply for the patent, and so that whatever you have, then you will be the owner, huh?
Yes, definitely, sir. Thank you, sir. Thank you for the suggestion.
Nice presentation.
Thank you, sir.
I could say. Thank you very much.
Thank you, sir.
Now you.
Okay. Next is Sayeda Nazida Sani, and the topic is, title of the topic, Multidisciplinary Center for Advanced Research and Studies. Right? This is the department choice, Jamia Millia. Topic is not given here. 8+2, right? Okay.
Good afternoon, everyone. I am Sayyida Najida Sani from Jamia Millia Islamia. My topic is CRISPR-Cas12a based detection of EGFR gene mutation in cell-free DNA for early diagnosis of non-small cell lung cancer. To introduce my topic, this is lung cancer. We already know that lung cancer is the most frequent cancer all over the world, and in India also. It is the leading cause of death due to cancer. Among the two types of lung cancer, small cell lung cancer and non-small cell lung cancer, non-small cell lung cancer is the most prevalent. It is around 80% cases in. And in main types of lung cancer is the non-small lung cancer, adenocarcinoma. Here we use EGFR gene for the diagnosis of this non-small cell lung cancer. This is the biomarker profile of the lung cancer, non-small cell lung cancer. From this we know that EGFR gene has around 35% prevalent, and in EGFR gene mutation, tyrosine kinase domain of the EGFR gene from exon 18 to exon 21, mutation is present in this exon region. For detection, we use cell-free DNA or light liquid biopsy and standard biopsy, tissue biopsy. For this, some challenges is there in the liquid biopsy, so we use liquid biopsy. It is reliable, it is non-invasive, less invasive, and it is we can use it for the therapeutic monitoring the therapeutic response. But in liquid biopsy, we have some challenges like low abundance of low level of mutation in the cell-free DNA. So we need sensitive detection methods like CRISPR-Cas system. We already know that there are many detection systems like EGFR. For EGFR gene mutation, we use next generation sequencing and RT-PCR, as we already know that gold standard for the diagnosis. In next generation sequencing, it has a high turnover time and time consuming. And in the RT-PCR also, we already know that it has a high false negative, and it is costly, and it needs technicians. And in case of so we are using CRISPR-Cas system, that is CRISPR-Cas12a, which can be used for the specific recognition, and above this we use RPA also, and then the lateral flow assay for the detection readout. This is the timeline for the CRISPR-Cas, which is used in the diagnosis. It is with the discovery of the CRISPR-Cas in the 1993. It has many applications like genome editing, and in the diagnosis also. We already know that Emanuela Carpenter and Jennifer Doudna, who has won Nobel Prize for discovery of the CRISPR-Cas9 for the genome editing. And in case of COVID-19 also, we already heard about the Sherlock detection method for the COVID-19. We know that in CRISPR, we are using CRISPR-Cas12a, which is a type of CRISPR-Cas system. It is a class 2 type A system. It is RNA guided endonuclease. We know the structure, which it needs. CRISPR-Cas12a has 3 components, like it is the CRISPR RNA and the DNA template and the Cas enzyme. With the recognition with the CRISPR RNA, it forms complementary with the template RNA, and it can recognize the target DNA. CRISPR-Cas12a has another property like collateral activity. It can cleave the target DNA, and again it can cleave any other DNA molecules which is present near the vicinity of the experiment. This is the hypothesis. We hypothesis that it can detect the cell-free from the cell-free DNA the mutation, and we use here detector mechanism for this. My objectives are design and validation of the various guide RNAs and RPA primers. And in the objective 2, we are optimizing the CRISPR-Cas12a based method for this EGFR gene mutation, and we will validate it through clinical samples. This is the schema of my work. This we will. We will amplify the target DNA with RPA amplification, and we will use the CRISPR-Cas system and the CRISPR RNA and the reporter molecules. It will, after recognizing with the CRISPR-Cas12a, it will cleave the DNA. After cleaving the DNA, target DNA, it can cleave the reporter molecule also. Thus, we can get the fluorescence. Also, we can detect with the lateral flow assay also. This is the result. I have prepared a enzyme. This enzyme, that is the protein LV-Cas12a. This is the SDS-based result of the induction of this protein, and this is the purification of this protein. And these are the target molecules I have used in this study. We synthesized the guide RNA, that is CRISPR RNA, for in vitro. This is the region for the guide RNA synthesis, and for detection we use in vitro cleavage reaction. We take the CRISPR-Cas12 protein and guide RNA and the target molecules and the fluorescence molecule which will be used for the detection. These are the result for the cleaving, showing the cleavage of the target DNA molecules with the enzyme, that is LV-Cas12a. These are the fluorescence region which shows a positive result, and with the help of the lateral flow assay also we can detect easily the positive mutation. We can detect, we can see the positive result here. If it is clipped, if it is clipped with the after recognizing with the CRISPR-Cas12a, it will cleave the reporter molecule, which is a fluorescence molecule sequenced by a after cleaving the sequencer part from these reporter molecules, it will give fluorescence. These are the conclusions. LV-Cas12a protein produced in-house. We have checked with cleavage activity. Guide RNA synthesized in vitro is also validated. CRISPR-Cas12a is validated for a type of mutation, that is exon 19 deletion. So we will optimize it with other mutation types also, and optimization of this is in progress, and we will take the patient sample for the validation of this experiment. So these are the reference, and I would like to acknowledge the authorities with this slide. Thank you.
I think it's okay. Is it okay like this? So this project is not yet concluded. Yes? Can you hear me? Yeah? So the project is not yet concluded.
Once part is completed, but we need validation by taking patient samples.
I see. Okay. Yeah.
The last but one, or you have one fluorescence. Okay, slide you have shown. These are what kind of fluorescence you have found? Is it the emission? Understand? Is it absorption? Or this is the phosphorescence? Can you tell me?
Is it fluorescence?
No, fluorescence is there. Is it the emission? Okay.
Yeah, it is emission.
Emission, and another emission is what we call it phosphorescence.
Yeah.
From this fluorescence and phosphorescence, which one is the one you have shown here? The slide.
Yeah, we use fluorescence molecules like FEM.
Yeah.
Here we use FEM.
PEM.
FEM molecules.
Okay.
Which is quenched by the quencher molecule after cleaving with the CRISPR-Cas enzyme.
One question. Okay, last. From that, you have found the fluorescence or phosphorescence. My question is or.
Fluorescence.
No. Then what is the time scale of that? If this you say fluorescence,
yeah.
Okay, how much second it observe this emission? Can you tell me?
Emission, we usually do it. We can detect in 15 minutes.
15 minutes is too much.
No.
No, the
actual
first
whole region.
Speaker, first you listen.
Okay.
The time which you have observed this fluorescence, that emission time, how much it is?
It starts with the cycle 1.
Okay, thank you. You are not doing that one. Yeah,
yes,
thank
sir.
you very much. Okay.
Okay, so our next speaker is Tanush Ridas. Okay.
Yeah. Slide please. Tanush Ridas. Sana was there. Sarvila was there.
Okay, a very good afternoon. A very good afternoon to everybody present here. Very good afternoon to all the respected dignitaries, to my fellow researchers. Today I am here to present my research, which involves the study the effect of excess zinc supplementation for a longer period of time in gene expression of antioxidant enzymes in Wistar rats and in their F1 generation. So by now, everybody in this hall must be very much acclimatized with the term trace elements. These are minute elements present in the living system, which is responsible for various physiological and biochemical processes. I have selected zinc as my research element. Zinc because it is an essential micronutrient which is responsible for growth and development of the body. So what happens, since zinc is very much responsible for growth and development, people tend to use it more frequently. It is used in agriculture and husbandry practices, it is used in baby food supplements, in multivitamin supplements. Studies have shown that the intake of zinc has increased day by day over the last decade and all over the world. So the question is, since zinc is a very much beneficial, it is non-toxic, it is beneficial, so excess of zinc, is it good or bad? Anything in excess is bad. So even though zinc is an essential micronutrient, excess zinc is discouraged by the health professionals. And the recommended dietary allowance of zinc varies depending on age, sex, and different physiological conditions. So what happens in our body, this metal deficiency and excess both promote oxidative stress, oxidative stress, and to counteract this oxidative stress, and our body has its own antioxidant defense system, which includes the antioxidant enzymes. So here comes our research question. What is the effect of long-term excessive zinc supplementation in the body? And if there is some effect, does this have any carryover effect onto the next generation? And nowadays we know that there is rising prevalence of different types of lifestyle diseases. So is there any connection between the excessive zinc intake with these diseases? So here is how we designed our experiment. We have taken 3 groups of rats, and we have fed them a synthetic basal diet, which we have designed. And we have fed them the diet for 180 days. And in the mid, we have also allowed them to mate for the F1 generation. So how we have divided, I will explain you once you understand how I divided the diets. So this is the basal diet, which I divided into group 1, group 2, and group 3. Group 1 has the control diet. Throughout the experiment, the diet composition was same, except in case of zinc, where in control diet it was given 20 mg zinc per kg. In group 2, 50 mg zinc per kg. In group 3, 80 mg zinc per kg. So the methodologies are, they were weighed every week. After the completion of the dietary period, blood sample and tissue collection was done. The blood was analyzed for the different biochemical parameters, and the tissue samples was for assessing the tissue metal status and different antioxidant gene expression. And all the methods were approved by the Institutional Animal Ethical Committee. So what happens in, we noticed that the body weight of the parent rats, it increased gradually. But after 150 days, there was a slight decrease in group 2 and group 3 on comparison to group 1, indicating that there must be some negative implication of excess use of zinc for a longer period of time. In F1 generation, there was gradual increase, so we can't say anything about F1 generation from the body weight, but from the parent rats, we can say higher zinc concentration in the diet and increased body weight of the rats were directly linked to increased food intake, which we will see in the next slide. So here the food intake also increased till 150 days, but after 150 days, there was a slight decrease. And also when we check the urine of the group 2 and 3 rats, which reacted positively with Benedict's test, indicating the presence of glycosuria in them. Zinc is highly potent in, thus we can say that zinc is highly potent in inducing observed state of metabolic syndrome that are not genetically predisposed to it. So when we check the biochemical parameters, there are different biochemical parameters. We found that group 2 and group 3 rats, there is a significant increase in the parameters in comparison to group 1, except in HDL, where there was decrease. And similar pattern was found in F1 generation of rats, suggesting that indicates a carryover effect of abnormalities associated with the parents to their offspring. So now we assess the metal level in liver and zinc. And what we found that the zinc level in group 1, group F1, 2 and F1, 3 increased significantly when compared to the normal, but the copper and magnesium level decreased. And the same trend was found in both liver and zinc, indicating the development of copper and magnesium deficiencies are related to zinc increase. It has also been reported, we did a thorough review and we found that the descendants of type 2 diabetes mellitus and different diseases, heart diseases, possesses higher zinc concentration and less copper concentration. And for gene expression study, these are the primers. And what we found that there is a significant downregulation in GPA, glutathione peroxidase, catalase, and copper zinc SOD. We have taken these 3 antioxidant enzymes because they are the first line of antioxidant defense. And these alterations are, these results are in accordance with our previous study by Sir, where he reported that reduced activities of antioxidant enzymes in rats on response to excess zinc supplementation. So basically, long story short, we can say that excess zinc supplementation for a long period of time disturbs the ionic balance of the body, and which thereby affects the proper functioning of antioxidant enzymes, as these ions form an integral part of these antioxidant enzymes, thereby altering the gene expression of antioxidant enzymes, resulting in oxidative stress to such a point that results in manifestation of different types of diseases. When we studied the gene expression studies of antioxidant enzymes in F1 generation, we found there is alteration, but there is non-uniform kind of alteration. So we concluded that this may be because of some post-oxidative stress, and because of the carryover effect of from parents because of zinc supplementation. This results open a wide, more deep study array where we can go do more deep study to come with a more robust explanation of this non-uniformities. So we can conclude that pharmacological dosage of excess zinc for a longer period of time have some negative implications in the parent rates and also in F1 generation. And the rising prevalence of cardiovascular diseases, even among younger generation, may be linked with this, with excessive zinc intakes. And this is the publication in a Scopus Index journal from the biochemical part of the study. And I want to thank everybody for your patience. Thank you.
Okay, again. You supplemented your animals to get this zinc excess. Is it very often in natural conditions to have zinc excess?
Sorry ma'am, can you repeat once?
Well, my question is, in natural conditions, without artificial supplementation that you used in your study, how often do you observe zinc excess?
We observe, there are studies of zinc to study the intake of zinc. But since the last decade, there were studies, and we found that the amount of zinc that people used to take then have increased because it is used. Zinc is used in every kind of multivitamin supplements, baby foods, agricultural and or animal husbandry practices. As a result, indirectly, they tend to take it more.
Okay, so the idea is that people who want to stay healthy take too much. Yes?
Actually, ma'am,
yeah, because you have the vitamin supplements.
Actually, ma'am, when doctors supplement this zinc concentration, they don't supplement it for a longer time. They supplement it for a particular time. If you have observed, they supplement for a particular time, and then they do tests to get any results. So for a very longer period of time, it is harmful. It should be stopped. Some alternative. It opens a very wide research area where we need to look upon.
I see. And another question, because for the animals that you used in your study, these were male animals. I mean boys, yeah? Rats.
No, we used Wister rats of male and female both.
I see. Because in the middle we have also assessed the effect in the F1 generation also. So we have taken male and female both. Yeah, because now there is a big stress on using both sexes. Okay, thank you.
One query is that, you have used superoxide. Can you see that?
Copper zinc superoxide.
Superoxide. Copper and zinc superoxide. Can you tell me the superoxide chemical formula? Because you are a biochemistry, that's why I'm coming to you. No. Okay. One more thing here, your work is very nice, fantastic, but you are using simply the copper, simply the zinc. Is it the 0 valency or 2 valency or 1 valency?
So the zinc sulfate, so you are, of the feed you are saying?
No, this you are using the copper, simply you are using copper. Is it the copper +1 or zinc +1 or zinc 0 valency? Sorry.
What's the chemical form?
I'm so sorry.
Okay. Next time when you study it, that is very important. From that point of view, interaction will start. Okay. Without knowing that the blindfold interactions happens. Okay. It's a very good. Okay. Congratulations.
Thank you so much, sir. I will take this into consideration. Thank you so much, ma'am.
My turn. Next is Serial number 14, Sheikh Benazir. Benazir, or how to pronounce?
Benazir.
Benazir. Okay, take your time.
Good afternoon. I'm Sheikh Benazir, research scholar, Department of Life Science, Botany. The topic of my presentation is physiological and biochemical responses of rice cultivars to drought stress at vegetative stage. So rice is a staple food of more than half of the world population. For the successful rice cultivation, it depends on various climatic conditions. So for the cultivation, sufficient water available is required. Drought stress is a major abiotic stress. Drought stress is a major abiotic stress which leads to drastic decline in rice production, and the salience of drought is even greater for rice when compared with other crops such as maize and wheat due to its relatively higher water needs. And in the previous research, it was mentioned that around 3,000 to 5,000 liters of water is required for the production of 1 kg of rice, which is about the double the amount needed for other crops. And so it is important to understand the salience faced by the farmers in the rice cultivation to ensure the sustainable production. And my objective is to determine the physiological and biochemical responses of the selected rice cultivars under drought stress and to promote the tolerant cultivars for future breeding program. Material and methods. For the experiment, the seeds were collected from the Wangbal Rice Research Institute, Hobal, Manipur, and one drought-tolerant variety was collected from the International Rice Research Institute for the standard. So screening was done. From the screening, 4 rice cultivars was selected, namely Sahau Pareton. Sak means rice, Hau means delicious, Sahau Pareto, which is black in color, Sahau Angangba, which is red in color, Sahau Angauba, which is white in color, Haitupau, and Sahavagidan. Sahavagidan is the drought-tolerant cultivar. So these are the rice grain of the selected cultivars. For the cultivation, the experiment was carried out in pot culture condition at Life Science Department, and first of all, the seed of its cultivars were incubated in water about 30 degrees Celsius for 1 day to facilitate the germination, and then the soil mix for rice plantation was prepared using field soil, and standard recommended doses of urea and superphosphate were applied, and the germinated seeds were sown in the 5 different pots and then allowed to grow for 15 days. Then before the transplantation, the soil-filled PVC pot were thoroughly watered and it was made into muddy and allowed to stand for 1 week before the transplantation of the rice seedling. The 15 days old plant were transplanted into the different pots of its varieties. And the phenolic content was quantified equivalent to gallic acid, and might be promote the plant damage.
Constituents of your rice. There was protein content, amino acid content. Yeah. These were expressed on the basis of dry.
Dry and the wet one. Which one is? I mean the dry and the wet, no? The proline content must be different, isn't it? Huh.
Yes.
The concentration must be vary because you're making is a microgram per ml, right?
Yes.
You said very simple, huh? Isn't it? Is there a little water content? You already said relative water is there. Definitely the concentration should be reduced. Understand? No? In the dry, it must be more. And one thing is that you are doing very much in the field of the soil. Okay, the same black rice. For example, it is you harvest here in the Kiamgai or this place. Okay, okay, Kansipur. You harvest in the Kaksing. Okay, and then taste will be similar or different. And the why it's so happen?
Different.
Huh?
Different. It
Difference.
might be due to climate.
Why it is? What is the significant reason behind?
Due to weather condition. Due to a change in weather condition.
Weather?
Or soil condition.
Yeah. Then you have any part of showing the pH scale here, the pH measurement
not
in this work?
showing here, but I did it.
Okay, I always suggest, no, please don't mind. Please you go always with the pH, right?
Yes.
Okay, from that significance, something will come, and the soil test TOC also required, no, to tell organic carbon, right?
Yes.
Yeah, that is very much needed for the the characterization or supplement of your work. Okay, thank you very much, presenter.
Okay. Okay, so the next presentation will be about stress of maize. And Rosina is the presenter. Rosina is the presenter. Okay.
Good afternoon, judges and all the audience present here. Today I'm going to present my research on drought stress in physiological and biochemical parameters of drought stress in maize tribal people of Manipur. So it can be said that maize is a staple food for this hilly region of Manipur. And the waxy corn that I use, they are of different varieties. We have the black, the mixed variety, which is yellow, red, black, and they are all mixed in this variety. Then red, cream, yellowish. These are the different varieties of waxy corn that are found in Manipur. These are the varieties that I use for my research. I collected the 5 after screening process. I have selected these 5 different waxy corn varieties from different districts of Manipur: Bishnupur, Senapati, Ukhrul, Thoubal. And Manipur is in a study done in 2021 by Council of Energy, Environment and Water. Manipur is ranked as the 6th most vulnerable state to climate change. And also in our state, agriculture is not very much well developed agricultural sector. And about 52.8% of our population is engaged as agricultural laborers. Because of this, it is high time we produce more high-yielding, drought-tolerant varieties which our farmers can use, and also to improve the irrigation facilities in the state. This is the method that I use. At 2-week-old seedlings were transplanted into these plastic pots that are filled with sandy loam soil of about 15 kg. The plants were allowed to grow under normal condition until pre-tasseling, that is 45 days after plantation. And after this, drought stress, 3 different levels of drought stress were given to the plants, while a group of control with 100% fill capacity was maintained for comparison. This is the plantation photo. Objective is to study the biochemical physiological impact of drought stress and to identify the most tolerant varieties. Relative water content of the different varieties are analyzed, and we see a decreasing trend in the relative water content of the variety, with 30 days stress plant being giving the least amount of relative water content. This is the chlorophyll content, chlorophyll A, B, and total chlorophyll of all the varieties, and we can see that the chlorophyll content decreases. The reason may be because of the disruption in the chlorophyll structure and allocation of lower assimilates to chlorophyll production in the plant. Now comes the osmolyte content of all the varieties: proline, phenol, protein, amino acids, and total soluble sugar. And as we all know, osmolytes, and also from the data that I have collected, osmolytes content of the varieties increase with increasing duration of stress. However, at 30 days treatment, the level of the osmolytes tend to decrease. The reason, I believe this chart shows the reason why osmolytes tend to increase under drought stress condition when stress is given to plant. The stress responsive transcription factors, they are activated. Transcription factors are activated, and the stress responsive genes, they signal the plant to produce more osmolytes such as free amino acids, carbohydrates, soluble sugars, polyols or alcoholic sugars, polyamines, and secondary metabolites such as phenols. They tend to increase under drought condition, and this is imperative from the data that I have found. Next is the antioxidant enzyme activity. I have analyzed superoxide dismutase activity and peroxidase activity, and we have seen that from this data the antioxidant activity of the plant increases with increasing duration of the drought stress in all the varieties. This is a chart showing the reason why there is an increase in the antioxidant activity. Under reactive oxygen species, as we all know, like singlet oxygen, then hydrogen peroxide, superoxides, they are generally present. They are generally a byproduct of plant metabolism. They are generally a product of plant metabolism, and they are involved in plant signaling also. However, under stress condition, the reactive oxygen species, the level of the reactive oxygen species tend to increase, and in this respect, plant increases the production of antioxidants such as SOD, then peroxidase, catalase, etc. to scavenge this increasing reactive oxygen species and become a drought tolerant plant. In conclusion, the study shows that under drought tolerance, the yield of the maize plant decreases. This is because in maize, the yield depends on the anthesis silking intervals, and the anthesis silking interval 3 days, and under stress condition, this interval is increased, and the yield of the plant decrease. And among the 5 varieties, variety V shows the most tolerance to drought stress, followed by W, N, and R. While
if we can go back up, superoxidase. Yeah, because well, there are more like this. There are several.
There are 2. Phenol and protein also I have written in my program.
Yeah, exactly. Yeah. Okay, so this is just a typo.
Yeah.
Okay, and then because well, you also express your data as on fresh weight. You heard the question that I asked to your colleague. Do you have any comment on this?
Actually, madam, we are analyzing the performance of plant at a particular stress level. We give the stress for 10 days, 20 days, and 30 days, and on that specific day, we tend to analyze all these biochemical data. So we did on fresh weight only, and we haven't analyzed for dry weight.
I see. Okay, so then your argument is that you analyze the plant as it is at this moment of the treatment.
On the day the treatment ends, on that day we analyze the.
I see. Okay, so it's more philosophical than.
Okay. Now you are showing here, Madam is asking the question about the unit. Madam is a very analytical.
Yeah, I am.
Okay. Purely analytical chemistry or technique. And here you can give the answer is, this milligram can convert into the gram or kg or ppm, understand? Parts per million.
Okay,
that you can give the answer in the very wise manner. Anyway, my question is the role of the phenol in your study.
Yeah.
Can you tell me?
Sir, phenol is a secondary metabolite, and also it is considered as an osmotic protectant also. So under normal condition, the phenol content is usually low, and when stress is induced in plants, the phenol content increases, so as to they also have take
say
part.
For example, phenol content is more,
Yeah.
and in your system, what it helps?
It helps in protecting the plant from drought stress, scavenging reactive oxygen species. It also takes part in that process also, sir. And signaling for more antioxidant production.
And another beauty word I want to listen from you, another word, just mention me and I'll be very happy. Like for example, I give you one example. This O3 is called ozone, right?
Yeah.
Ozone layer. Ozone layer is a protective of many things. And similarly, these phenol have a very major role to protect from your plantation. Okay,
yes.
That is what one ray is there. Can you name it? One ray is there. Can you name it?
In phenol, sir.
Yeah, in phenol, no, it has a very important role in your plantation. Okay.
Yeah.
Anyway. Yeah, you are going to the ROS or something, you go. Please, last question.
Reactive oxygen species.
Yeah. Yeah, here, one is you wrote one, superscript, O2, subscript. What it means?
Sir, this is O2 is...
Yeah. If you can give answer, I'll give highest mark. Anyway, very nice presentation, of course.
Thank you, sir.
But whatever you touch it,
yes.
Please you try to make understand.
Yes.
Little in depth.
Yes.
Okay, thank you very much. Nice presentation.
Thank you. Now we shall go for lunch, and we will reassemble back at 2:30 to continue the rest of the presentation. The lunch will be at the courtyard of our Department of Zoology. Thank you. Please make sure that you come back by 2:30.
Thank you.